Chapter 12 covers the definition of a species, how species develop and reproduce, and also has a discussion on human races. Scientists have a hard time agreeing on what constitutes a species. There are different concepts that can be used to define species, but the most commonly used one is the biological species concept. In the biological species concept, a species is defined as a group of individuals that can naturally interbreed and produce fertile offspring, but cannot reproduce with members of other species. In practice, this can be difficult to apply, as some life forms can reproduce asexually, and we often don't know how fossilized life forms reproduced. In the morphological species concept, a species is defined as a group of individuals with some reliable physical characteristics that distinguish them from all other species. However, because of the wide variety in some species between male and females, even this definition has some problems. When we look at the fossil record, we typically apply the morphological species concept to that record to kind of figure out different species based off of their fossils. The genealogical species concept describes species as organisms that can interbreed and are all descendants of a common ancestor and represent independent evolutionary lineages. This concept requires detailed knowledge of the genes within a population, so until we've identified the genome of a lot of different organisms, it can be difficult to apply this concept as well. We also can't use this concept for species in the fossil record since we don't have their DNA and can't figure that out very easily. The total of all the possible alleles found in all the individuals of a species is that species' gene pool. The spread of an allele throughout a gene pool is called gene flow. This occurs when reproduction is occurring within a species. If two subgroups of a species are isolated from one another, there could potentially be changes to the subgroup's gene pool because certain alleles might no longer be available to one of these two groups. If the environmental pressures are different between the two subgroups, this could eventually lead to distinct changes between the two subgroups to the point that they might be considered different species. This process is called speciation. A particular trait becomes more common in a population due to environmental pressures. That also means the allele for that trait has become more common. You could also say the frequency of that allele has increased. The allele frequency is the percentage of copies of any given gene that are that particular allele. The Hardy-Weinberg theorem is a mathematical formula that's used to calculate allele frequencies in a population. This theorem states that allele frequencies will remain stable in a population and therefore can be calculated if the population is large, if the population is randomly mating, and also if the population is experiencing no migration or natural selection, so if the environment is fairly constant. Here's the Hardy-Weinberg equation. In this equation, P is the frequency of a particular dominant allele in a population, and Q is the frequency of the complementary recessive allele in that same population. For example, if we're talking about the trait of height in a pea plant, which you've already done Punnett squares on before, P would be equivalent to the frequency of the uppercase T allele, so the dominant allele for height in the population, which would be tallness, and then Q would be equivalent to the frequency of the lowercase t allele in that population, so the allele for shortness. P squared is the frequency of the homozygous dominant individuals, which is indicated by two uppercase T's if we're talking about the height of a pea plant. Q squared is the frequency of the homozygous recessive individuals in the population, which would be indicated by two lowercase t's if we're talking about pea plants. And then 2pq is the frequency of the heterozygous individuals in that same population. So if we're talking about pea plants and height, that would be the frequency of the individuals that have the genotype without a dominant T and a recessive T. If we know the frequency of the homozygous recessive individuals in a population, we can then figure out the frequency of the recessive allele. So in this example, the frequency of the homozygous recessive individual is 25%, or 0.25, and that would be equivalent to Q squared, in the equation. So if you take the square root of q, or the square root of 0.25, that gives the value for q, which is the recessive allele in the population, the frequency of that recessive allele. There are only 2 alleles possible in the population with this particular combination. So if the frequency of one of them is 50%, then the frequency for the other one would also be 50%, because 50 plus 50 equals 100. So if you put those values back into the equation, you can figure out the frequencies of the homozygous dominant and the heterozygous individuals in the population. If given enough information, you should be able to use this equation to find the frequencies of either the alleles or the individual genotypes in a given population. Here's another example. So Rh factor is a protein that's found on red blood cells in some humans. We talked about it in one of the previous chapters. Rh positive individuals make this protein and are dominant to Rh negative individuals who don't make the protein. So if you're given the frequency of the Rh negative individuals, which would be the ones that don't make the protein, which would be the recessive alleles, the homozygous recessive individuals, if you're given that, the percentage of that in the population, and let's talk about maybe Dallas, and this is a made-up equation, this is not actually true, but this is made up. So the frequency of the Rh negative, or homozygous recessive individuals in this population is 16%, then what will be the frequency of the heterozygous individuals for this trait in Dallas? Well, we know that q squared, which is the frequency of the homozygous recessive individuals according to the equation, we know that that is 16%, or 0.16. So the square root of that gives us q, which is 0.4, or 40%. There are only 2 alleles for this trait, so if the frequency of the recessive allele is 40%, then the frequency for the other allele, the dominant allele, must be 60%, or 0 points. That means that p, according to the equation, is 0.6, so p squared would be 0.6 times 0.6, which would be 0.36, or 36%. Plugging those numbers back into the equation gives us a way to double check our values, and yes, that does equal 1, so we're correct with our calculations so far. So 2pq is the frequency of the heterozygous individuals in the population, which is what the original question was asking for. So 2 times 0.6 times 0.4 gives us 0.48, or 48%, and that would be the frequency of the heterozygous individuals in this particular population would be 48%. So whenever you're looking at these questions and you're using this equation, remember there's a difference between the frequency of just the dominant allele and the frequency of the recessive allele, totals 100% when you add them together, versus the frequency of different genotypes. So the frequency of the homozygous dominant, the frequency of the heterozygous individuals, and the frequency of the homozygous recessive individuals, because all 3 of those groups would also equal 100%. And so you can use that to kind of play around with the numbers and figure out the value that you're looking for if you're given enough information for that equation. A change in an allele frequency that occurs due to chance is called genetic drift. It tends to occur in 3 different situations. In the founder effect, a small sample of a larger population is going to go away and establish a new population. In this new population, there is a smaller gene pool than in the original population, so the frequency of the alleles is different in the new population. In a situation where there's a small population that chooses to interbreed amongst itself, as with humans, such as in a religious sect, certain alleles that may only be found in 1 or 2 individuals could gradually die out if those individuals did not reproduce. So that's kind of the second example of genetic drift, which would be a small population. In the third example, which is sexual selection, that occurs when males and females within a population have preferences for particular physical features in their mates. These preferences can cause populations to differ in appearance, leading to allele frequencies over time.